So in the AACR 2021, I presented my work entitled Overcoming Immunosuppression and Therapeutic Resistance to PARP Inhibition in BRCA1-deficient breast cancer with STING agonists. PARP inhibitors have drastically changed the treatment landscape of advanced ovarian cancers with BRCA mutations. However, the impact of this class of inhibitor is relatively modest in patients with advanced BRCA mutant breast cancers. For instance, although PARP inhibitors significantly improved progression-free survival, the final reports of clinical trials suggest no overall survival benefit for both olaparib and talazoparib in patients with breast cancer, highlighting the need to understand why some BRCA mutant breast cancers are refractory to PARP inhibitors in the effort to develop strategies to improve the responses to PARP inhibition therapy. Our understanding of the mechanisms underlying the therapeutic efficacy of PARP inhibitors is still evolving. Since first described in 2005, PARP inhibitors have been shown exert synthetic lethality in BRCA-deficient tumor cells. Recently, we and others demonstrated that in addition to the synthetic lethality, the immune responses triggered by PARP inhibition is also required for the effective anti-tumor response of this class of drug. For example, in PARP inhibitors-sensitive ovarian cancers, PARP inhibitors lead to release of double-stranded DNA by tumor cells, which activates stimulator of interferon genes called STING in the intratumoral dendritic cells, thus triggering a type 1 interferon response and subsequent induction of anti-tumor C8 T cells. In the current study, we developed syngeneic genetically engineered mouse models of BRCA1-deficient breast cancer and ovarian cancer that recapitulates both the genetics and the pathology of advanced BRCA1-deficient tumors in patients. Using those mouse models as well as human cell lines and clinical specimens, we uncovered a major mechanism by which BRCA1-deficient breast tumors typically progress through treatment with the PARP inhibitors, in contrast to the much higher rates of sepsis observed for BRCA-deficient ovarian cancer. Our major findings are number one, BRCA1 mutant breast tumors are refractory to PARP inhibition in vivo in immune-competent mice. PD-1 blockade does not add therapeutic benefit to PARP inhibition. Number two, BRCA1 mutant breast cancer cells render macrophages pro-tumorigenic or M2-like phenotypes and functions both in vivo and in vitro, which is independent of PARP inhibitor treatment. Number three, tumor-associated macrophages suppress tumor cell DNA damage and cell death in response to PARP inhibition, resulting in reduced production of cytosolic double-stranded DNA and synthetic lethality, thereby inhibiting STING-dependent activation of dendritic cells and macrophages. Number four, STING agonists reprogram M2-like macrophages into M1-like anti-tumor macrophages in a macrophage STING-dependent manner. Number five, STING agonists restore anti-tumor immunity to the tumor immune microenvironment in vivo, thus sensitizing BRCA1-deficient breast tumors to olaparib. Number six, STING-deficient breast tumors fail to respond to the combination of olaparib and a STING agonist administered intratumorally. However, they respond effectively to systemic delivery of STING agonists in combination with a PARP inhibitor. Targeting pro-tumorigenic M2-like macrophages has been actively pursued in the field of cancer immunotherapy with various agents, such as small molecule inhibitors or monoclonal antibodies against CSF1, CSF1 receptor, TGF-beta receptor to deplete or suppress macrophages. Many of these agents have been evaluated in early phase clinical trials with little success. Here we show for the first time that M2-like macrophages suppress olaparib-induced lethal DNA damage and abrogate STING activation, and that a STING agonist is able to effectively reprogram M2-like macrophages to M1-like anti-tumor macrophages. Thus, a STING agonist can not only restore the synthetic lethal response to PARP inhibition, but also reshape the tumor immune microenvironment to promote immunogenic anti-tumor responses that synergizes with PARP inhibition in BRCA mutant breast cancers. In addition, our demonstration that STING activation in immune cells is sufficient to elicit anti-tumor immunity suggests a novel therapeutic approach for treating a significant fraction of patients with cancers deficient in tumor cell intrinsic STING. Hence, the translational implications of these findings are highly significant as they provide a strong and timely rationale for combining systemic administration of STING agonists with the PARP inhibitors in the treatment of BRCA-deficient breast cancers, particularly in light of two recent publications in Science reporting anti-tumor activities of two non-nucleotide STING agonists that can be administered systemically. We hope our mechanism-based novel therapeutic approach to circumvent the refractory nature of BRCA1-deficient breast cancers will provide instrumental in guiding the design of clinical trials evaluating PARP inhibitor-based rational combination therapy in the near future.